Hey, Greysteel Nation, Sully here with the Barbell prescription and another research review, an important one, I think. I'm joined again by our friend, colleague, and research review faculty, Victoria Volkov from Tel Aviv University in Israel. Good evening, Vic.
Good evening.
Evening where you are. Thanks for joining us again. I know you're really busy right now, and I should mention that Vic found this paper for us, and I think it's really cool. I think it's exciting. So we're going to take a quick look today at Aguiar et al. Master athletes have longer telomeres than age-matched non-athletes, a systematic review, meta-analysis, and discussion of possible mechanisms. This is in Experimental Gerontology 2021. I always love it when the authors put the answer right there in the title, as you know. And that title sounds like a big deal, and it is. So, Vic, before we get into the paper itself, you want to tell us a little bit about telomeres?
I'll do my best.
All right.
So, as much as we know, well-accepted marker of biological aging is the telomere length. Telomeres are actually repetitive DNA sequences, TTA, GGG, at the end of chromosomes, and they are important to maintain the stability and integrity of a genome. Cell division leads to telomere length shortening, that via cell malfunctioning in turn may impair the rate of cell division in tissue. This is a natural process. It happens during aging, and eventually it leads to loss of tissue function, and it is characterized as biological aging. We know that any lifestyle factor that protects telomere length is thus potentially starving off the aging process and may well lead to increased number of healthy life years. So we know that telomeres are stabilized among the rest by the sheltering complex that protect the telomeric DNA. We'll talk about it later in the discussion. And of course, pro-inflammatory cytokines and anti-inflammatory cytokines, they have a role in telomeric length. Thus, any factor other than the sheltering complex that counteracts these pro-aging agents can also protect the length during aging. In this context, we know that physical activity has indeed been associated with longer telomeres. This is something that has been studied for the last decade pretty much, and this is what we're going to discuss now.
Very good. Yeah, so almost everybody who ever talks about telomeres, including the person who discovered them, talks about the shoelace analogy, like they're the cap at the end of the chromosomal shoelace, and it gets, every time a cell divides, it gets shorter, as you said, this telomere section, and eventually you run out of telomere, and so before the whole thing can unravel, the cell says, well, that's the end of me, and basically commits cell suicide. So it's promotes a cell suicide or apoptotic process. I was thinking about this paper when I woke up this morning, and I was thinking, the genome is rolled into chromosomes. These telomeres at the end are the chronosomes, right? The time bodies, the actual molecular embodiment of time and aging. So like the length of the telomere is the sand left in the cellular hourglass, is kind of how I should say it. Now, I should point out that Vic found an excellent video, a TED Talk by Elizabeth Blackburn, the scientist who won the Nobel Prize for her work on telomeres. And if you want to find out more about them, and you do want to find out more about them, this is a good place to start, and we'll link that video in the doobly-doo for you all. So again, this is a meta-analysis, which is a study of studies. It's where they went out and found a bunch of relevant studies and windowed them down into studies that could be combined into meaningful data, and showed us what the best literature on this topic seems to show at this time. And I don't think we need to belabor the methods too much here. I thought this was a pretty well done systematic review and meta-analysis. They followed the Cochrane Handbook, the PRISMA and Moose guidelines for studies of this type, and they used pretty good eligibility criteria. They looked at all observational studies in masters that compared telomere length in any type of tissue sample, including blood, muscle, and not saliva, but buccal swabs. They excluded animal studies, which I get, but they also excluded randomized control trials and studies where athletes were classified according to activity level. And I'm not sure I get that. Maybe you can help me out with that later when we discuss it. The search strategy was pretty standard, as was their data extraction and quality assessment of the studies and stats. So they went out, they got the papers, and then they assessed them for their methodology and various other quality issues. They looked at heterogeneity and bias risk of the incorporated studies, as they should, as well as a methodological assessment. So their initial search pulled up almost 11,000 papers, 4,724 after removal of duplicates. And out of all of those, they found 11 that met their inclusion and exclusion criteria and could be used for meta-analysis and review. Those 11 studies incorporated 240 masters. So this is like a study of 240 masters and 209 age-matched controls. And it's important to point out that they're talking about competitors here when they're talking about masters athletes. And I know that you and I will have more to say about that later. Telomere length was measured in a variety of samples and by multiple methods, including Southern blot, PCR or polymerase chain reaction, and by direct visualization with fluorescent in situ hybridization, which gives you really pretty pictures. There was, as we might expect, heterogeneity in study quality and risk of bias. And if you want that level of detail, you should go ahead and pull this paper and the individual studies. 4 of the studies were found to be at moderate risk of bias, 7 at low risk. The incorporated studies didn't all just look at telomere length. They also looked at underlying mechanisms like the oxidative stress or free radical stress, which can damage and shorten the telomeres, as you pointed out, and expression of the sheltering complex, a complex of proteins that protect telomeres from accelerated shortening. So Vic mentioned this earlier in her introduction of the telomeres. The sheltering complex is this, it's 6 proteins, but there's a bunch of other proteins that are associated with the sheltering complex. It's a very complex system, but basically think about it as a protective cap on the shoelace that helps protect the shoelace from damage that might result in accelerated shortening. So again, it's a meta-analysis, not an original observation, and I think it's fairly well done. I don't have any major methodological concerns with the meta-analysis itself. So Vic, can you tell us what they found?
Okay, so first of all, we know that the purpose of this meta-analysis was to assess whether master athletes have longer telomeres than age-matched non-athletes. And they also wanted to discuss the possible underlying mechanisms that lead to the preservation of telomere length in master athletes. And there, in all of the 11 studies, master athletes were people over 35 years of age.
A little young. Yeah.
Little young, yeah, and there was heterogeneity in the ages also, not only in the age, in the master athletes population, but also in the control samples. And we'll get to it later. But as to the results, when we look at the telomere length in general, then we found that master athletes had longer telomeres than age-matched controls. And it was significantly significant difference. And also they had longer blood and buccal cells telomeres than the control group when they looked at the different places where they took the measurements from. So this we can see in the figure.
Yeah, it was interesting. They found longer telomeres in blood cells and in buccal cells from the cheek, but not really in muscle cells. That was interesting.
And skeletal muscles.
Right.
Yeah.
And we can talk about why that might be so a little later. Yeah, go on.
And then we wanted to discuss that lower oxidative stress and chronic inflammation, as we know, with a higher expression of shelterin proteins that protect the telomeres, may contribute to the longer telomeres. So this is what we're going to look in the underlying mechanisms. And what we see as to the oxidative stress, that 3 of the selected studies found an association between oxidative stress parameters and telomere length in master athletes. One was classified as moderate risk of bias. The other two were at low risk of bias. So this is also mentioned. One of the studies demonstrated that master athletes that participate in competitions from 100 meters to marathon running have longer telomeres and lower levels of a byproduct of lipid peroxidation, which is T-bars, and also higher antioxidant defenses, as reflected by higher catalase, SOD, which is superoxide dismutase, and also the ratio between SOD and T-bars. And all this when compared to untrained age-matched individuals. Similarly, the report was that master endurance runners not only have better oxidative balance and longer telomeres than age-matched non-athletes, but these were even better than in the young untrained individuals, which was very interesting.
Very interesting finding, yes.
And these observations apply to both sprint and endurance master athletes by the year of 2020, the study that was done, the latest study that was done in the meta-analysis. And endurance athletes had lower levels of all the biomarkers of oxidative stress, catalase, Trolox equivalent, and others, and higher levels of superoxide dismutase and nitrates, which means the oxidizing agents. So this was
the
antioxidant.
Yeah. So
superoxide
dismutase is an antioxidant enzyme that converts superoxide into hydrogen peroxide, which can be easily removed by catalase. So it's a good thing. It's a good thing to have more superoxide dismutase, right? Yep.
Yeah, we'll see it in the mechanisms chart. So lifelong training seems to be a key point to improve oxidative balance of master athletes by mitigating biological aging and the onset of chronic diseases, providing a healthy and functional trajectory. This was their go-to after this result as regarding the oxidative stress. Another mechanism that was discussed was chronic inflammation. So 3 studies addressed the effects of lifelong exercise on pro-inflammatory cytokines and telomere length in master athletes. The interesting findings here, in one study, endurance athletes had higher levels of inflammatory cytokines. We're talking about STNFRI, ileoleukin-6, and ileoleukin-15, and they had it more than sprint athletes. While sprint athletes had higher levels of anti-inflammatory cytokines.
Interesting.
Which, IL-10, IL-10 ratio, and TNF-alpha, and also the ratio between ileoleukin-10 and ileoleukin-6. So the sprinters had a better anti-inflammatory cytokines profile than endurance athletes. Also, the last study reported negative correlation between markers of chronic inflammation and telomere length, and a positive association between telomere length and the performance level of master athletes. So this evidence suggests that exercise is beneficial to reduce or attenuate the inflammatory status in older age, as we know. So what can we say about it in practical terms? So far, the literature indicates that lifelong training, they call it training. We know it's probably because we're talking mostly about aerobics of master athletes. It has a beneficial effect on the balance of pro and anti-inflammatory cytokines. And
we're also
therefore,
I'm sorry to interrupt. We're also looking at, actually, I thought it was interesting that the sprint athletes actually had less inflammation than the really aerobic athletes. I thought that was interesting. It also says something about intensity.
And they also mention it when they're talking about dose response relationship further on when they conclude the mechanisms. So all these things may attenuate inflammation, immunosenescence, and thus the risk of age-related chronic diseases. This was the conclusion here. As to the mechanism of sheltering proteins, so we also already talked about it. We know that these proteins protect telomeric termini, the ending, like you said, the shoelace caps, from triggering any DNA damage response and then an apoptosis of the cell. And So the findings suggest so far that higher physical fitness may have a beneficial effect on sheltering complex and proteins and telomerase activity. Because only one study here that was classified as moderate risk of bias investigated telomere length and its regulatory proteins in master athletes. So they talked about all the sheltering proteins complex, and also CHOC2, mRNA, P53, and
various
P16.
protein markers of apoptosis and apoptosis.
Of apoptosis, yeah.
Yeah.
So this was a single study, but it did find that the aged athletes showed down regulation of P16 and greater expression of Ku70 and Ku80 mRNA, which is good. And the integrative perspective of all the mechanisms suggested that although the current literature does not provide experimental evidence so far to fully establish all mechanisms that underlie the preservation of telomere length in the master athletes, of course, compared to non-athletic controls, there are several potential mechanisms here. One is lowered oxidative stress, which we talked about by increasing antioxidants and antioxidant defense mechanisms. Second is reduced chronic inflammation. And third is a higher activity of telomerase and protein-related integrity of telomeres. So this was the integrated perspective,
which I thought was pretty good, and it definitely worth a read on its own. I thought, yeah. If you look at the graphical analysis of the data in figures 3 and 4, you can see that the aggregate data on just on telomere length across the studies was pretty clean. It was pretty consistent. The data on oxidative stress and antioxidant capacity was a little bit more mixed, but the trend still seems pretty clear there as well. Only 3 of the studies looked at chronic inflammation, which could affect and be affected by telomeres, but wasn't really enough to do an actual meta-analysis per se, and not enough to really point to a strong correlation, in my opinion, although I think that's probably right. I think it's probably there. We do know that inflammation is important. We even have a term for it. You used it earlier, inflammaging. And we know that exercise and diet are important here. But I'm not sure that there's enough yet to draw a firm line between the inflammation and the telomere length. So we see this increased telomere length in masters athletes, and we see the decreased inflammation in masters athletes. But is that a direct cause? Is there some sort of direct molecular causal mechanism that's there? My suspicion is that there is, but I don't think any of the data here necessarily proves it. The shelter-in proteins, which play a key role in telomere maintenance, and which form this complex of 6 proteins that are in turn associated with this web of other protein interactions, is a very interesting field of study. But they only had one paper that looked at it. Not enough there for a true meta-analysis on the question of exercise and shelter-in specifically. Although again, I think that they're probably onto something. So there was that one study that suggested better shelter-in expression and the telomerase activity. So we've mentioned the telomerase several times. That's the enzyme that sort of, not necessarily repairs, but extends that telomere cap. So you definitely want that to be active under the right circumstances. So this is a huge and important area that's just right for study, and the authors make some really good suggestions about the right way to go forward. How future studies should look at this, because this is a big deal. Anything else in terms of the results or the discussion before we get into our own discussion? What this all means. Yeah,
they also mentioned dose response. Exercise connection in master athletes, which was it's important to talk about a little. So we know that although master athletes are recognized as successful model of successful aging, a small number of athletes may experience some kind of under recovery or overtraining condition known as fatigued myopathic athlete syndrome, which is called briefly FAMS. So they did say that this condition may be due to endurance training at very high volumes and intensities. We know that several of the studies were conducted dealt with ultramarathoners and people that had a large amount of weekly volume. So
and they ended up with shorter telomeres.
And they ended up with with shorter telomeres and also chronic inflammation syndromes, oxidative damage.
So too much of a good thing. Like any other medicine, you can overdose it, right? So the dose is the poison, as Paracelsus once said. And so I thought that was a very interesting point. How directly does it feed into this whole discussion of intensity versus volume? I don't know, but I think we can say that you can overdo it, and that's
especially when you're competitive. Yeah.
Especially when you're competitive. So let's open our open discussion with that. So we found here, or they found here, as you point out in this paper, that athletes have longer telomeres, and that it may be due to differences in oxidative stress, shelter and expression, telomerase activity, and possibly inflammation as well. And they found increased telomere length in blood cells, white blood cells, macrophages, stuff like that, buccal cells, basically, the cells from the mucous membrane inside the cheek, but not in muscles. And I thought that was interesting. They didn't find much difference between, the older master athletes and young athletes and age match controls. Why would that be? And I think it would be because skeletal muscle is not a very mitotically active tissue, right? So it's just not, your muscle cells are not, it's not like, your blood cells or your skin cells or the cells in your gut. It's just not a tissue that's undergoing a lot of cellular, what do I want to say? It's not a tissue that's undergoing a lot of cellular turnover. And so I wouldn't expect to see that. The other thing that I thought we wanted to discuss was the issue of how they defined athletes. So this is a population of competitive athletes. That's what they're talking about when they talk about masters athletes. And what I take away from that is competitive, the population of competitive athletes was the best way for the investigators of the various papers that went into the hopper of this meta-analysis to get at a well-trained older population. Right? So these were people who engaged in competition. They were engaged in training, right? Actual training as opposed to just intermittent exercise. So this was a good population for them to study. What are the implications for older athletes, for older individuals who are engaged in training, but not necessarily competition? I think it's the same. Training is training. So I don't think that the practical implications of this are limited to people who are engaged in training at the competitive level over 35 or 40 or 50. I mean, do you have anything to say about that?
I think it might also be the reason why they excluded RCTs, because they were dealing with competitive athletes, they could not interfere with their program. So it was easier to observe. And as you said, they as active as a master athlete can get.
Yeah, that's a good point. That hadn't occurred to me. I think that's a really, really good point that you make, because I'm like, why would they exclude RCTs? That's a good point, a very practical point. And I think you may have nailed it there. The other thing I wanted to discuss was in the video that we're linking to, the video that you found, Elizabeth Blackburn is talking about the role of telomeres in senescence and aging. And at one point she makes this point, so it's great. So all we got to do is pop our little telomerase pill and increase the activity of our telomerase, and we're good to go. We're gonna live for 1,000 years, just like the immortalized pond scum cells that she initially characterized as telomeres and telomerase activity in. But it's not that simple, is it? We can't just, you know, overactivate our telomeres by some sort of pharmacological or, you know, molecular medical approach and call it a day, because there's a dark side to this too, isn't there? So when it comes to immortalized cells, that's not necessarily a population of cells that you want to have in your body, right? Because that's one of the characteristics of many types of transformed or cancer cells. So if you look at a lot of cancer cell lines, for example, the HeLa cell line, they are immortalized cell lines. And I don't know about HeLa in particular, but a lot of these immortalized cell lines have high telomerase activity, and their telomeres never shorten, and they just keep growing and growing and growing. So it's another great example of a biological process that you would like to exploit to your advantage for good health and healthy aging. But the way to do it isn't necessarily by directly upregulating telomerase or shelterins or something like that, because you can get into trouble that way. The best way to do it is to do it, you know, the natural way. So because we know that masters athletes don't have higher rates of cancer. In fact, quite the opposite is what the data seems to show, and yet they have longer telomeres and appear to have more activity of telomerase. So this is, you know, this is the way to do it, is through lifestyle. And both the authors and Elizabeth Blackburn talk about several lifestyle factors, including exercise, that are going to promote longer telomere length, and stress reduction has also been implicated. Good sleep has been implicated, and so on. So what's the best way to do this? Well, the way that we're discussing here is through exercise medicine and lifestyle medicine. The point that you brought up about overtraining, volume, excessive competition, and possible telomere shortening, I think that bears mentioning again, as well as the issue of high intensity stimulus that was mentioned by the authors. Now, you know, I like that. You know, a high intensity dose of exercise medicine. The authors mentioned that there needs to be adequate intensity to see this biological effect. But as much as I like that, I can't say that they actually presented any data here to demonstrate that or even to support it, except for that one thing that you mentioned. It would be extremely interesting to do some sort of stratified observational study where we looked at, you know, endurance athletes versus resistance athletes, strength athletes versus aerobic athletes, and looked at the differential expression of telomerase and the length of their telomeres and so on, but that has yet to be done. And while we're on that topic, I think it's also important to point out that we can't state a hard and fast cause and effect relationship from this data. This is all highly correlative, right? So what we take away from this paper at the end is that there's definitely a correlation between training and increased telomere length for people of a certain age. And that's exciting, and that's good. But I don't think we're in a place yet where we can say, in fact, I know we're not in a place yet where we can say that increased training promotes increased telomere length in aging. We can't say that, you know, that it runs in that causal direction. Maybe increased telomere length, maybe people with longer telomeres engage in training, as opposed to people
maybe they were born this way.
Maybe they were born this way, and people who are born this way are more likely to be active in old age. So we can't say, you know, which direction the causal arrow goes, if there is a cause and effect arrow, but we can make inferences. And I think it's pretty reasonable. You know, do you really think it's the case that the population of masters athletes overrepresents those who are genetically predisposed to have longer telomeres? I find that a little bit more difficult to swallow than the speculation that people who engage in training after their 30s have longer telomeres. I hope I'm making that clear. I think it's far more likely that the cause and effect relationship flows in that direction. And it makes sense from a sort of teleological point of view too. Like when you engage in activity of a certain intensity and volume, and you remain active, and you engage in training at a level that's going to support competition or healthy aging, that you're sending a signal to your body that, yeah, I'm not done yet. You know, we need to keep those telomeres long because I'm still out here. I'm still hunting and gathering and reproducing. I'm still vibrant and alive. And, you know, so let's hit the snooze button on those telomeres and keep things going a little bit longer. That makes more sense to me. But there's no proof here of that. So I think it's very exciting.
They were sincere about it.
What?
They were sincere about it.
Yeah, they were. Yeah, to their credit, they were very upfront about that. Like we have not established a cause and effect relationship here. We've just established a pretty strong correlation, and it's really, really interesting. And we're starting to scratch the surface on the causal mechanisms. Again, like I said before, I think that the integrative analysis that they gave us of, you know, how this might all work, and there's a beautiful figure. Figure 6 here. So
they did brought up an interesting question. At the end of the discussion, they thought about what happens to telomere length in case of cessation of the competitive activity.
That's a study.
The mastocytic, yeah, it's an idea for an.
That's a study that needs to be done. Yeah. So what happens? Yeah, detraining and telomere length. That would be a very interesting study. And you know, that would not be a terribly difficult study to do. You know, absolutely. And it would give further support to this idea that there's a causal relationship between the activity and the telomere length in that direction. And some of Elizabeth Blackburn's own work also supports that too. I mean, we can look at various stressors or lacks of stressors in our lifestyle and in the environment. And when she and other people have looked at that kind of thing, she talks in the video about people who are caregivers for Alzheimer's patients, I think it was, for example, people who are under chronic stress and they have shorter telomeres. And so, you know, I think that just adds credence to the whole idea, and this is what we're really talking about with this paper, that our lifestyle affects the rate of aging. We talk all the time about how exercise has these systemic benefits on, you know, obviously muscle mass and strength and bone density and mobility and balance and cardiovascular health and insulin sensitivity and metabolic health and visceral fat and chronic inflammation. I mean, we've looked at that literature and all that stuff is there. That's all kind of systemic, right? The actual sort of phenotypical changes that we see from using exercise and lifestyle medicine. This is data that gets right down to the very core. I mean, this is the nuclear, genetic, molecular level of what's going on here, and I think it's pretty exciting. So what we are seeing is potentially the effect of exercise and lifestyle medicine at every level of biological organization, and that's exciting to me. How many medicines have an effect at every level of biological organization? That's pretty cool. So, so take that and do with it as you will. I think the practical implications are pretty clear. You know, keep your telomeres long, people. Long telomeres are good. You have anything to add, Vic?
I think that to the common reader of this study, question that comes in mind, if I want to start now a prescribed weekly training program as a novice, even from scratch, when will I see, what's the minimum effective dose? When will I see the benefits of my telomere length preservation? And we cannot answer it, of course.
We cannot answer. We don't have that data yet. So some young investigator like you needs to go and find out. So thank God we have people like you in the world who are interested in studying these kinds of problems. Because I will tell you one thing for sure, Vic, I'm not going to do it. I did my time at the bench. It's over. But yeah, I mean, if you're a young investigator and you're interested in exercise medicine, you know, there's 6 or 7 or 10 or 12 PhDs, you know, their doctoral dissertations outlined in this paper right here. So young people, young scientific people, go out and get it, because there's a lot of exciting questions here. Vic, great job, and a great pickup. Thank you for finding this paper and bringing it to my attention. I think it's potentially important. It's a great addition to our database, and I appreciate you taking the time out with me to break it down for our listeners.
Thank you.
And I know you have lots of homework to do, and so I'm going to let you go. All of you, thanks for watching, and until next time, stay strong and stay healthy. Bye for now.
Bye-bye.